Hello, today we are going to learn gram staining. Gram staining is one of the most important and widely used differential staining technique in microbiology. So, objective of our today's learning first to learn the gram staining technique and using gram staining technique. Secondly, we are going to differentiate between gram positive and gram negative bacteria. So this is a widely used differential staining technique. Gram staining, it is discovered by Kristen Gram in 1883. Gram staining technique have taxonomic importance in identification and classification of bacteria. It differentiates bacteria in two groups as gram positive or gram negative, depending upon whether they retain or lose the primary stain crystal violet when subjected to the treatment of decolorizing agent like alcohol. This staining technique required various reagents like primary stain, for example crystal violet, mordant like grams iodine, decolorizer that is alcohol, and a secondary stain like safranin. The difference between the gram positive and gram negative bacteria, it is mainly depend on the cell wall composition of that bacteria. M. J. Sultan proposed a hypothesis based on cell wall composition for gram staining mechanism. The gram it is mainly depend on the lipid content and peptidoglycan content in the gram negative and gram positive cell walls. The lipid content in gram negative cell wall it is 10 to 20%, whereas in gram positive it is only 1 to 2%. Hence, when we apply the primary stain crystal violet, it forms the complex with the mordant grams iodine to form a CVI complex. And when the cell treated with the decolorizing agent, the lipid from the cell wall of gram negative bacteria it is extracted, which results in the increased porosity of the cell wall. Okay, which leads to the escape of crystal violet iodine complex from the gram negative cells, and the cells appear colorless on the treatment of decolorizing agent. Now such decolorized gram negative cells could take the secondary color safranin and they could appear as pink in color. In contrast, the cell wall of gram positive bacteria, because of their low lipid content, become dehydrated during treatment with alcohol. So the cells shrink in size, their porosity of their cell wall decreases, and the CVI complex, that is the crystal violet, the violet color, retain inside the gram positive cells, and even after decolorization treatment, the gram positive cells appear violet in color. Not only lipid content but peptidoglycan content also contributes for this gram staining mechanism. Peptidoglycan content of gram negative organism is only 5 to 10%, whereas in gram positive organism it is 40 to 90%. So in case of gram positive bacteria, the CVI complex, it is trapped into the cell wall on the decolorization treatment because of the high peptidoglycan content. As the decolorization treatment, the alcohol treatment carrying out shrinkage of pores into the peptidoglycan layer. And as in case of gram-positive organism, higher peptidoglycan content is there, more shrinkage in the pore size is carried out. So the porosity of the cell wall decreases, and whatever CVI complex present inside the cell, it retain inside the cell, and the gram positive cells appear violet in color. Whereas in case of gram negative organism, due to the high lipid content, porosity increases, and due to the low peptidoglycan content, the shrinkage of the peptidoglycan will be carried out at lesser extent. So ultimately the porosity of the cell wall, or in case of gram negative, on the decolorization treatment, it is quite larger, sufficient to drain out CVI complex, and that is why on the decolorization treatment the gram negative cells appear colorless, which could retain the secondary stain safranin. So we move towards the procedure. Hope you will understand up till now. Yes? Okay. So in the procedure, the very first step is prepare smear using bacterial suspension on a clean grease-free slide, air dried, heat fix it, and during this procedure, so you up till this procedure you will get all the cells colorless. Then cover the smear with primary stain crystal violet for 2 minutes. So on the treatment of crystal violet, all the cells, whether they are gram positive or gram negative, they appear violet in color. Next step is to wash the slide with gentle running tap water. Then cover the smear with mordant grams iodine for 1 minute. This grams iodine forms a CVI, that is crystal violet iodine complex, with the primary stain crystal violet. Even at this stage, both the cells, gram positive and gram negative, they appear violet in color. Again, we should go for a gentle wash of the slide under gentle running tap water. Why we are going to use the gentle running tap water? To avoid washing out of your smear. After that, apply decolorizer alcohol in dropwise manner to the smear until the violet color stop to come out. Now difference is made at this stage. As we discuss in the mechanism, your gram negative cells, they have high peptidoglycan content and low lipid content. On the decolorization treatment, the porosity of the gram positive cell wall, it is quite lower, quite less. It does not allow washing out of the CVI complex, and that is why the cells, they appear violet in color. Whereas in case of gram negative cells, the lipid content is high, peptidoglycan content is less. So on the decolorization treatment, the porosity of the cell wall is more. sufficient to drain out the CVI complex, and hence the cells appear colorless on the decolorization treatment. After decolorization, again we have to wash the slide with gentle running tap water. Then cover the smear with secondary stain safranin for 5 minutes. Now at this stage, as your gram positive cells, they retain the CVI complex, they enable to gain that secondary stain and they appear violet in color. Whereas the colorless gram negative cells after decolorization treatment, they acquire the secondary stain safranin and appear pink in color. After that 5-minute treatment of safranin, pour off the stain, wash the slide with gentle running tap water, blot dry the slide, then air dry the stain smear and put a small drop of immersion oil and observe the slide under immersion lens, oil immersion lens. The purple color cells, they are the gram positive cells, whereas the pink colored cells, they are the gram negative cells. So how you could interpret the result of gram staining? The organisms which retain violet color, which appear violet in color, they are gram positive organisms. Whereas the organism which retain the pink color, which appear pink in color, they are the gram negative bacteria. Yes. So examples of gram positive organism, that is Bacillus, Clostridium, Cornybacterium, Streptococcus, Staphylococcus, Lactobacillus, Propionibacterium. These are the some genus belonging to the gram positive organisms. Likewise, gram negative. Could you guess? Yes, the Pseudomonas, Escherichia, Klebsiella, Acetobacter, Proteus, Shigella, and Salmonella. These are the examples of gram negative organism. So up till now, we discuss about the history, introduction, the requirements of reagent in gram staining technique, procedure of gram staining technique, the mechanism behind how the gram positive organism appear purple and why the gram negative organism appear pink in color. We discuss all the things. Your questions are welcome. So outcome of our today's learning is how to perform the gram staining. So at the end of session, you know how you can carry out the gram staining technique and how you can differentiate between the gram positive organism and gram negative organism using gram staining technique, which could be utilized as the taxonomic studies of the bacteria.